Hello, everybody. Today, I'm going to be talking about value realization in laboratory automation, how we can identify and achieve attainable goals. I work for a company called Synthego. It's located in the San Francisco Bay Area in California in the United States. If you browse to our website, you will see our mission statement, which is displayed here on the slide. Synthego is a genome engineering company that enables the acceleration of life science research and development in the pursuit of improving human health. At Synthego, we believe that we can industrialize life science. People can serve to our site and order CRISPR gene editing kits, as well as engineered cells. We believe that life science will be advanced by a combination of biology and engineering disciplines. We focus on CRISPR gene editing, which is a relatively simple, cheap, and precise technique for genome editing. The figure on the right shows the principle behind CRISPR. A guide RNA guides a Cas9 protein to a target area on the genome. The Cas9 protein causes a double-stranded break, indicated by DSB in the figure. This DSB can cause a gene knockout, meaning that the gene is no longer functional. Or alternatively, in combination with some donor DNA, a gene knock-in happens. For example, to repair the functionality of a gene. At Synthego, we fabricate both guide RNAs for our customers who want to do the editing themselves, but we also provide a service of engineered cells on demand. So our core business is to provide cell editing as a service so that our customers can focus on the consequences of the edits. For example, to study disease models, drug response, or to develop cures for genetic diseases and cancer. This is very similar to ordering electronic circuit boards from a vendor. You don't really care about how the circuit board is being made or how the chips work. You really care about the functionality of the board. We want to enable this paradigm in biology. So my name is Arne Vandenbroucke. I'm the Director of Automation and Systems Engineering at Synthego. And on the right here, you see a picture of my team members. These people have backgrounds in physics, biology, chemical engineering, electrical engineering, robotics, and mechanical engineering. Industrializing biology is not necessarily simple or easy, and we believe that we need multidisciplinary approaches to tackle these problems. The team's mission is to establish and support process automation across Synthego, both using commercially available equipment, as well as building and validating in-house hardware. Let's talk a little bit about laboratory automation. When engaging on lab automation, we often want the moon on a stick. We want fully automated processes that can run with as little as possible supervision and that can run day in, day out. However, achieving that final goal can be challenging. It can be a long and windy road towards that end goal. And if you don't anticipate that the road is lengthy, the process can be a bit frustrating. So if you only focus on the end goal, we will have challenges. I would like to point out today that we can create value along the journey. When talking about value creation, this cartoon here really says a lot more than 1,000 words. If we want to build a car, but we start with a wheel and then a chassis and then perhaps a body and then ultimately a car, we will only create value at the end. The journey is long and windy, and quite frankly, the probability to come up with a car is rather low. You might come up with something else than a car. If, on the other hand, as indicated by the bottom half of the figure, we start by building something that already brings us faster from A to B, like a skateboard, for example, we do create some value. Maybe not much, but it's something that we can immediately use. If next we build a bicycle and then perhaps we add an engine to keep adding value, We keep creating value as we develop the final product. This is also indicated by the happiness of the smiley face in the figure. Ultimately, we may never end up with a sedan or like the car we initially designed, but we have created value nonetheless along the way, while every time becoming more efficient in what we want to achieve, in this case, moving from place A to place B. So even when we don't completely achieve the end goal, our customers, and whether that's lab operators or senior managers, can be happy. So intermediate goals may look like they are far off from the end goal, but that doesn't matter. Each of the intermediate goals add value, and it's important to identify the value created within the iterative steps to keep going and to achieve ultimately the final goal of full automation. So I'm going to highlight 5 different topics, and the first topic is that automation brings process standardization. Whenever we are going to automate processes on lab equipment, we will force ourselves to define our processes to an intricate detail. When that process, or even part of that process, is automated, we will reduce operator dependency, and we will enforce the same starting conditions. And this is how we can achieve standardization and have a much larger chance of having repeatable and predictable processes. And as an example, I would like to highlight our capability of producing engineered cell libraries. So this is one of the products we offer at Synthego. ECL, Engineered Cell Library, is a library where we knock out every gene that is important for a process, one at a time. And the intended result is a microtiter plate of cells with a different gene knocked out in each well. For example, we can knock out the immune system genes to study the efficacy of certain drug therapies. One of our customers requested to knock out 1,300 genes with 2 replicates, so that is 2,600 edits in total. We then also needed to stimulate a response in each of the wells and collect the cell supernatant. Obviously, for these amounts of edits, we'd like to use automation, and we did. And although it wasn't the moon on a stick, I believe we did achieve significant value by standardizing the process. Let me first give some background in how CRISPR editing actually works. The first thing that we need to do for CRISPR editing is to make the guide RNA Cas9 complex. This is also known as ribonucleoprotein, RNP. Then secondly, we need to deliver the RNP inside the cell nucleus. And for that, we use a process called electroporation, where we apply an external electric field across the cells. That external electric field forces the cell membrane to open so that the RNP complex can enter the cell. Once the E field is gone, once we turn it off, the cell membrane goes back to its natural state, but the RNP, the guide RNA and Cas9, will be inside the cell. And so editing can happen. Now, in the lab, what we need is we use microtiter plates. We add Cas9 protein and guide RNA inside the plate together with a transfection buffer. We also add cells into that plate. Now, this could be a regular plate. But to do transfection, we need a dedicated transfection plate indicated on the right here in the figure. This is a plate where each of the wells is covered with electrodes so that we can apply a field across each well. As mentioned before, we use automation to aliquot these reagents. For the low volume transfers, we use the Beckman Echo. And to dispense a transfection buffer, we used a manual pipette. As I said, not everything was automated for this process. The cells were aliquoted using a Giger Certus, and the 384 plate stem from the regular 384 plate into the transfection plate was done using Hamilton star. Next, we bring the transfection plate to an electroporation device. That device actually zaps 384 wells sequentially. While transfection is ongoing, a recovery plate is filled with recovery media using a biotech EL406. And after transfection, cells are transferred to the recovery plate using a Hamilton star. We put these devices in an automated work cell within an ISO 5 enclosure. The ISO 5 reduces the probability of contamination. That could be bacterial or fungal contamination. So for this process, we use the Hamilton liquid handler, the Lonza 384 well nucleofactor, Beckman Echo, Biotech dispenser, and the Combi dispenser. I'm going to show a little video of how the plate is being brought to the nucleofactor while the recovery plates are being prepared. The nucleofactor is in the lower right corner of the video. That plate is being presented now. We discard of the lid and we bring it to the Biotech device to dispense recovery media. Then that plate is brought to the Hamilton deck. Of course, you didn't see the Hamilton deck. That's in the next video. Here we see how the transfection plate is being brought to the Hamilton deck, and then the Hamilton robot will move that plate internally so the transfer can happen. This was our first massive screen. So remember, 2,600 edits. And the process needed a lot of hand-holding or also scaffolding, as you can see an example of scaffolding in the picture on the right. It was actually supervised by the entire team. Not everything was automated. Not all dispensers were integrated with the work cell. We had the manual addition of the transfection buffer, and we had to prepare the Hamilton deck manually. But nonetheless, by using the automation equipment, or even partial automation equipment, it required us to minutely define the process. And so we achieved standardization. And so if our next customer would come along with a similar large engineered cell screen, we would be ready, and we could actually iterate on top of this design. Next, I would like to highlight how automation can avoid bottlenecks in the process. Talking again about this engineered cell library assay. Once we have the edited cells in 384 well plates, the task at hand is to expand these into 96 well plates. The customer actually wanted to do that twice to create 2 technical replicates. What the customer then also wanted is to induce a response in the ECL, the engineered cell library. And after we induce the response, measure the plates in a plate imager within 24 hours. So we had 8 384 well plates of edited cells. That went into 64 96 well plates. Now, if in all these plates we need to induce a response and image it 24 hours later, when imaging takes about 10 to 15 minutes, you could see that you can create a bottleneck. To make a case here, I'm going to use some slides that were generated by Hi-Res Bio. What we see here on top is a Gantt chart of actions going on with 4 assay plates in a system where we have a liquid handling deck with 4 shakers and 4 thermal cyclers. So before any action happens, we need to wait until all assay plates are loaded. So there is an amount of waiting time at the beginning of the thermal cycling, and there's a waiting time at the end. This is called batch processing. If, on the other hand, we go to streaming, where a robot arm would feed plates to a thermal cycler that is no longer on deck of the liquid handler, but somewhere in the work cell, we see that the processing time for each of the plates is now uniform. Thus, this streaming approach accounts for process unification and avoids bottlenecks. All plates are treated identically. And to make a case even further, we can go to an extreme example. If we go up to 100 plates, we can see that streaming significantly reduces the time. But more importantly, it allows for process unification. There is no variable waiting time between assay plates. We apply the streaming process to the ECL screen, where we had different processes. The first process was the transfection, and process 2A was the expansion from 384 into 96, and the second process was the imaging of those 96 well plates. So here you see the process in red, orange, yellow, and the two greens for 5 transfections. And we could arrange it so that we'd never had overlap between the imaging processes 2B. The third point, I'd like to highlight how automation also allows for process miniaturization. And bringing it back to CRISPR, we can talk a little bit about CRISPR quality control. Now, quality control is typically done with DNA sequencing, where we read back the genome that we edited. Next generation sequencing provides the best sequencing quality and is cheapest when doing many edits. NGS also has the advantage of being massively parallel. We combine many different targets into one sequencing run. And in our case, we combine 1536 samples into one vial. Of course, we need to label these samples, but that's part of the NGS library preparation workflow. Once we have this one vial with the 1536 samples, that vial can go into the sequencing instrument. In our NGS reactions, we start with cell lysis that gives us genomic DNA. We then perform 2 PCR reactions, one to amplify the gene of interest and one to add barcodes to the region of interest. After these 2 PCR reactions, we do a size selection procedure using magnetic beads. Now, importantly, each of the PCR reactions use PCR primers and master mix. Both these are actually expensive reagents. So if we can reduce the reaction volume, we can reduce cost, but at the same time also increase throughput. In order to achieve these smaller reaction volumes, we have to use precise liquid handlers and liquid dispensers. The Certus and Mantis devices are great low-volume dispensers that yield good precision, and we use these for master mix and magnetic bead dispensers. The Echo is really good at small volume dispense, but also at individual well-to-well transfers. Its any well-to-any well capability is really unique to the instrument, and we use the Echo to transfer 100 nanoliter of primers. Transferring 100 nanoliter by hand is, I would say, impossible. And then as the bulk liquid handler, we use Formulatrix Fast, which is a relatively new device. Bulk is probably not the right term here, since we use only 1 microliter transfers, but the bulk refers to the fast stamping or 96 input plates to 384 PCR plates, and also copying the output from PCR1 into a PCR2 plate. We also use the fast for elution. So again, automation here allows to use smaller well format plates, 384 plates, smaller reaction volumes, and smaller volume transfers. And this is a picture of our NGS process that we have today. We don't have fancy robots yet, but that's sort of the point. We use walk-up instrumentation that allows us to use process development. But while we use the walk-up instrumentation, we use equipment tailored for low volume handling, so we already get those benefits. Next, I'd like to talk about how automation can enable sophisticated DOE experiments. Design of experiments, DOE, allows to study many factors and their interactions simultaneously. And to give a little background on design of experiments, we can consider this graphic here that I took from the reference mentioned on the slide as well. Let's say we have an experiment where we want to optimize the response as a function of temperature and concentration. What we could do is vary the temperature first on the x-axis. Let's say we have 6 measurements indicated by the blue crosses, and one of them gives us the best response. That's the response indicated by the thin red circle. Once we have the optimal temperature, we can then decide to vary the concentration, again with 6 measurements. And within those 6 measurements, we have an optimum indicated by the thicker red circle here. However, this is a false optimum because we have ignored the dependency between temperature and concentration. Instead, if we vary concentration and temperature simultaneously, we are able to identify the surface describing these 2 parameters and also be able to identify the optimal point on this surface indicated by the red circle. And let's look at electroporation and what factors influence the transfection process. There's actually many factors. Could be the guide to Cas ratio, the cells to RNP ratio, the type of transfection buffer, the electrical pulse pattern. Let's say we want to set up such an experiment, and because it's biology, let's say we want 4 replicates per point. Note that, of course, some DOE designs allow you to get away with less replicates, but just for the sake of arguments, let's just assume we want 4 replicates that we put in a PCR plate. If now we also want to study 3 different ratios of guide to Cas, we would utilize 3 columns of the plate. If now we also wanted to evaluate 3 different pulses, we would be using 9 columns. And let's say we wanted to study 2 different buffers, we would be using 18 columns. Note that here we have a so-called full factorial design. There are others, perhaps more optimal designs, DOE designs, but that would be beyond the scope of this presentation here. Imagine, though, you had to execute the experiment by hand. This would be tedious and error-prone, as you need to be very careful what mixture goes where. And even worse, there is no randomization. This means that this experiment is actually potentially subject to systematic row or column effects. Ideally, you want to randomize the distribution of the parameters across the plate. Luckily, we can use automation to enable these sophisticated experiments. We can use dispensers, like the Beckman Echo, to dispense guide RNA and Cas9 protein. We can use other dispensers, like the Mantis or Certus, to dispense different amount of cells. And these dispensers can also be used to dispense different buffers. And when using automation, we can also program the Alonso nuclear factor automatically. This still might be daunting to translate these DOE designs into robot instructions, but fortunately, some companies provide dedicated software that help translate these DOE designs into robot instructions. And one of these companies is Synthego. They have, for example, DOE for assay development. You can definitely Google them if you want to know more information. Now, lastly, I'd like to highlight how automation results in proper data and metadata storage. One of the tasks at hand in cell engineering is monitoring cell growth. Percent confluency is the amount of well area that is covered by cells. On the left image, there should be one cell somewhere. It's hard to see. But on the right, you can see clearly that we have a large number of cells. Once we have a large number of wells with high confluency, it is time to expand from, let's say, a 384 well plate to a 96 well plate. We monitor the percent confluency using an imaging cytometer. Imagine having to do this by hand. You would load a plate in the instrument that would give you a data file, but you would have no context. Context is typically referred to as metadata. What cell line do we have? How many days after transfection are we imaging? What are the imaging settings that were used? Now, when running automated experiments, metadata can be easily captured, again, due to the need for having systematic processes. We will know exactly how much time has expired since the last transfection. So proper data storage has an added benefit. It also enables data mining and machine learning models. This is the work cell where we monitor cell growth at Synthego. Plates run 24/7 in this automated work cell. It's also an ISO 5 environment, and they cycle between incubator at 37 degrees C and imaging systems. Just to give you an idea about the throughput, since January 2021, we have processed over 6.6 million images. These images are automatically uploaded to AWS, Amazon Web Storage, and contextualized with info from the barcode reader. So it's the barcode reader that can probe information about what cell is in that plate. Here is a little video about this work cell. we see on the upper left a plate being ejected from the plate reader. That plate is oriented from the landscape into the portrait position, and then it's being brought into the incubator that you can see on the lower right in the picture. The incubator retracts the plates, and it will provide a new plate after some time. So I've tried to highlight 5 benefits of automation. And there's one more point I'd like to discuss, is how we can achieve scalable automation solutions using APIs. API stands for Application Programming Interface. That's a software intermediary that allows 2 applications to talk to each other. A client, for example, a laptop, can get something, put something, post something, or delete something from a database. And this interconnectivity is crucial in building out an automation facility. And this is a trend observed in the last few years only, that major vendors have recognized the needs for APIs in life science, and many come up with APIs into their software. As an example, let's consider GBG, Green Button Go, which is Biosero Scheduler. Evidently, the scheduler already has API capabilities into instrumentation, using .NET client wrappers or using gRPC. But the third bullet point in the slide here says new for the REST Web API. The blue box shows what we can do with these remote API commands. We can load a process, start and stop a process, get status of a process, all remotely, show hide GUI, get set variables, execute certain scripts, query data tables, handle errors, subscribe to events. These are very useful if you want to do remote monitoring or remote control of your processes. Also, Hamilton, which is a liquid handling vendor, has recently come up with a vector API that allows to load methods, start and terminate runs from a remote client. And this API is new as of 2020. And so let's talk a little bit about why APIs get so much attention. The reason is that we don't want monolithic protocols. For example, in our imaging scenario, we don't want one imaging order where the scheduler would have to sit idle for 6 hours. Instead, we want to keep it nimble, and we want another program to launch just the imaging program. The other program would be responsible for the waiting time. So APIs actually allow that process to happen. So we can have another application in the middle of the figure here with the business logic that can submit orders to the work cell or to the liquid handlers. But these business logic programs can also interact with laboratory information management systems. The business logic can actually be written in a relatively lightweight app in Python. Furthermore, if we expand on this model, we can also have data processing programs that interact with the entire application. Now, while this may look really complex and maybe people in lab automation aren't quite used to APIs, there's many external third parties that can help. There's Zavo R3 and Mosaic that can help with laboratory inventory management systems. TetraScience can help with data processing. Artificial is a company that can help you write your business logic around controlling your work cells or liquid handlers. But also, very importantly, this structure is actually a standard software engineering structure. So we can leverage the richness of software engineering that exists outside of life science into life science. Cloud applications have grown in general in software engineering in, let's say, the 2010s, and I'm very excited that in the 2020s we see that technology being brought over into life science. So this brings me to the summary. I hope I could show you today that automation helps you to bring standardization, avoids bottlenecks, allows process miniaturization, enables sophisticated DOE experiments, and results in proper data and metadata storage. Even if you do not achieve full automation, these value items can be created along the way. Like even and the cartoon indicates the point. When we want to build a sandcastle, even if we start with a little mound with a flag on, it's already something, and the kids can be happy. I've also highlighted how scalable automation solutions can be built using APIs. Thank you very much for your attention.